Okay, so now we're going to start chapter 7. So in chapter 7, we're going to move quickly through the lymphatic system. So basically, when you take a look at the lymphatic system, you're taking a look at another vascular system, so similar to the cardiovascular system with a few differences and a few specific nuances associated with it. But in the lymph system, you're going to have lymphatic vessels and you're going to have specific organs as well. Some of the capillaries, the job of a lymphatic capillary is to basically absorb interstitial fluid. So that's that fluid that's in your tissues as it goes through the capillary. So it basically mops up excess fluid that keeps fluid from being stored in your interstitial tissues, keeping you from swelling. They also, another main job that they have is absorbing fats. Lipids is basically how you absorb lipids. So in specific types of lymphatic capillaries called lacteals. And then, of course, you have lymphoid organs, which typically are part of the immune system. And so as a result, these are going to be associated with carrying and providing structure for your white blood cells, so basically the cells of your lymph system. Now, in the lymph vessels, you're going to see a fluid called lymph. And so lymph capillaries, unlike vascular capillaries, are a little bit different in the sense that lymph capillaries are not closed. So capillaries in your vascular system, in your blood vessels, are closed. So they basically go from the heart, they go through the arteries, through the capillaries, through the veins, back to the heart again. And so your blood should never leave the blood vessels. But the lymph system, the lymph capillaries are open. So this is an open system. So it's an open circulatory system. And so as a result, then typically what happens is lymph will be collected from your tissues. So basically, if you take a look at a lymph capillary, it looks something like this. So the tissues will have fluid in them, and then the fluid will basically get collected like a big funnel into the capillaries. And then from the capillaries, it'll start to go toward the cardiovascular system and through a couple of ducts, the main ducts, the thoracic duct and the right lymphatic duct. These basically will drain into the cardiovascular system. So the thoracic duct will collect lymph from the upper portion of the body, from your thorax, at your chest area, your left arm, the head and neck regions. And then the right lymphatic duct will return lymph from the right side of the arm, not the left arm, but the right side of the head and neck. So you have it kind of divided up into right and left halves of drainages. So this is kind of what it looks like. So if you kind of look closely here, you'll see you have all these lymphatic vessels scattered throughout your body. And so they're basically collecting up fluid from your tissues, bringing it back through these major duct areas. So you can see your right lymphatic duct region, which is going to be right in this area right here. And then you can see the thoracic duct, which is going to be this main big one right here, that kind of drains blood from kind of goes up and back and around behind the heart. So you can't see where it goes up. Kind of the heart is in the way, but it basically goes up and then back and around. And you can see how it basically tethers to the left side. In this case, the brachiocephalic and goes back to the heart. So it basically dumps fluid into your bloodstream as it returns to your heart. There are a couple of other organs associated with the lymph system. For instance, your tonsils are part one of the lymphoid organs associated with your lymph system. The thymus, which typically in adults is mostly turned and converted into kind of fatty tissue. Your spleen is also part of your lymphoid tissue. And so these kind of all have a little bit of that same sort of effect, especially that immune support effect. So typically when you take a look at the larger vessels, they're very similar to veins. And so, for instance, they'll have valves, just like we learned about in chapter 6, where you have, 5, where you have basically valves inside your veins that prevents the backflow of blood. In this case, you're preventing the backflow of lymph. So it's the same basic idea. In this case, lymph is being moved because of your skeletal muscle contraction. So remember, that was one of the 3 mechanisms that we had for moving venous blood back to the heart. And so this is the dominant method for moving lymph back to the heart itself. So when you take a look at the lymphatic organs, you have a couple of main types, right? So you have what's called primary, or the ones that are kind of more central to the system, and of course, it's secondary. The primary organs are typically associated with the main lymphatic role, which is like the immune system. So you have the red bone marrow, which is the site of new blood cells, white and red. And then, of course, you have the thymus, which is where you develop your white blood cells for your immune system. The secondary lymphatic organs, which are not secondary in importance, but secondary in organization, are the kind of the peripheral organs, if you will. So those are the lymph nodes, spleen, and the tonsils. Those are the ones we typically interact with kind of structurally. And so here's kind of what it looks like. So here's your red bone marrow. So you can see that in your spongy bone, you're going to have, and we'll talk about bones later on in the semester, but this is basically where your white blood cells and your red blood cells are being made. Your thymus, where you have these large sort of structures, these cortical and medullary structures that are designed to mature your white blood cells so that your immune systems can have highly trained and well-trained cells. And of course, your spleen, which is harboring not just white blood cells and red blood cells, but is also a major site of red blood cell breakdown. Then, of course, your lymph nodes, right? So this is kind of like an army barracks associated with your white blood cells. So you'll see both B cell and T cell lymphocytes, and we'll talk more about those a little bit later, being housed here. So you can kind of think of the lymph node as sort of an army barracks or a military base, where you have kind of like a presence throughout the body. And so you'll notice that throughout the body, you have all these different lymph nodes, which you can see here. So all these little individual swellings, these little puffy parts, these are the major lymph nodes spread throughout your body, the army barracks, essentially, of your lymph system, so that you can always have a ready supply of white blood cells ready to go on the attack if you get invaded. So the red marrow, we'll start off with that one. So this is basically blood cell production. And so in children, most of your bones have red marrow. In adults, some of that turns into fat. And so we only have a little bit of red marrow. We don't need quite as much because we're not actively growing like children are. And then when you take a look at what's happening in the red marrow, not only your white blood cells developing here in your red blood cells, but specifically white blood cells called the B cell lymphocytes. These guys are maturing here. This is the reason why they're called B cell lymphocytes, is because B stands for bone. They mature and they're ready to go in the actual bone structure itself. In the thymus, which is another primary lymphatic organ, this is a structure that rests right on top of the heart. And typically it'll have a couple of different functions. Number one is it produces a hormone called thymosin. And this is basically driving the differentiation of another type of lymphocyte, which are the T cells. The T cells are, and the B cells basically compose some pretty major white blood cell groups that are associated with your immune response. Typically, immature T cells are made in the marrow, and then they're released so they can go to the thymus where they mature, which is the reason why they're called T cells, T for thymus. And so these will mature in the thymus, and then they'll be released to basically protect the body. So for the secondary organs, we have the spleen. The spleen will filter your blood, and typically it's composed of what we refer to as white pulp and red pulp. And in here we have lots of macrophages. These are basically phagocytic cells that will do all kinds of stuff. So these are the guys that will gobble up bacteria when they invade your body. They'll also gobble up debris if you happen to be injured. And they will also recycle worn out red blood cells. And so all of those are functions of the spleen. And so the spleen itself then being an organ with some of that reticular tissue in it, will have an outer capsule. And so it has a fairly thin outer capsule. So if you happen to hit it really hard, you can rupture the spleen and they'll have to remove your spleen. Now let's take a look at lymph nodes. So these are the army barracks, basically, that kind of intersect almost like rest areas on a freeway, the different lymphatic vessels. And so these guys will filter the lymph. They typically have a lot of connective tissue in them and a capsule around them that kind of divides them all up into sort of these cubicles or these regions or compartments. They tend to be filled with macrophages. Again, you're going to be phagocytic cells that are engulfing pathogens and debris as they come in contact with the lymph nodes. So basically, if you have an invader and it makes its way into the lymph nodes, that's kind of like walking into the enemy's army barracks. They're going to basically kill you and destroy you. And so you have your lymphocytes in here, which are essentially the major cells that fight against infections and cancer cells, along with macrophages. So these are all very important cells and structures in the lymph system. And so generally speaking, when you take a look at lymph nodes, we will oftentimes name them. There'll be names for each of these lymph nodes, which you're not going to need to know, but there'll be names for each of these, and they're named for the regions. For instance, if you have lymph nodes in your armpits, which is something that when you have bubonic plague swell up, and so you get these big buboes or blue type swellings in your armpits. Those are the lymph nodes in your armpits that are swelling up. Those are referred to as the axillary nodes, because the axillary region is your armpit region. Inguinal nodes are the ones in your groin area. Those are typically the ones we worry about. If somebody has testicular cancer, we worry about the cancer spreading into the inguinal nodes and then spreading to the rest of the body from there. So we'll have common neck lymph nodes that we have in front. So like, for instance, if your lymph nodes are swelling, you'll be able to feel them in the front of your neck, and of course in your groin and armpit, just like we mentioned. Lymphatic nodules are smaller structures. So these are like lymphoid tissue. They don't have a capsule, so they're not like a fully grown up lymph node. So these are just smaller, like little aggregations of lymph tissue. So the tonsils are lymphoid tissue, and they have the same function as lymph nodes. So basically, they'll fight off infection. They're located, typically outposted with your respiratory tract. So basically, anything that you're inhaling or ingesting will be attacked by the tonsils. And then you have another type of tissue called the Peyer's patches. This is basically found in the intestines, in the intestinal wall, and their job is to fight off any infections that come via what you eat in your digestive tract. And so you also have Peyer's patches also in the appendix as well. So the appendix is actually lymphoid tissue. So it's not useless. It's actually part of your lymph system. Okay, now let's take a look at the immune defenses, and this might be able to bring some clarity to some of these cells. First of all, when you take a look at your immune system, we can kind of break our immune system. So what we have is a system that's designed to kill and remove foreign things, pathogens, cancer cells, things like that, abnormal and foreign things. There's two major branches. You have innate and you have adaptive. So the innate is typically a fully functional branch where you can do some attacking without, you don't have to be exposed to a pathogen to do this one. It's already there. The adaptive is only initiated when you're exposed to a specific pathogen. So you adapt to a new threat. So oftentimes the innate was referred to as non-specific because essentially it attacks without recognition. And so generally speaking, some of these non-specific types of defenses that you'll see come in a couple of flavors. First of all, barriers. You'll have both physical and chemical barriers, and we'll talk about these here in just a little bit. You'll also have what's called the inflammatory response. This is more of like a chemical type of response, a non-specific attack. On an invasion or something that doesn't belong in the body. And then you have lots of different types of protective proteins, things like something called complement, which is a group of proteins that attacks bacteria. So they don't recognize anything. So they don't recognize a pathogen. They don't have memory of it. They can't identify, this is E. coli. That's not the way they work. They just basically know that it doesn't belong there and they go on the attack. And so here's kind of like some of the pieces. So you can see you have the barriers to entry. That's one of your innate defenses. But when that gets breached, you typically have exposure of mucous membranes and things of that nature. Then you'll have different types of protective proteins that will essentially answer the call. So those are basically in your bloodstream. Complement proteins, for instance, and interferons. These are all basically types of immune attackers, if you will, that will attack different types of invaders, different types of pathogens. And then the phagocytes, like macrophages, in this case, we use natural killer cells. These will basically come to the site of infection. They'll start killing these bacteria. So for instance, you can see here's a neutrophil. This is a type of phagocyte. The natural killer cells are a type of phagocyte. The dendritic cell is a type of phagocyte. So you can see they're gobbling up these bacteria and they're destroying them, all three of them acting together, going after them. This is a macrophage here. So all three of these guys are phagocytic killers. They engulf the bacteria and they chop them all up with their lysosomes. And then, of course, you have cytokines, which are basically chemical messengers. And these guys are designed to coordinate the attack and communicate with the rest of the immune system of what's going on. And then you get your inflammation response from this. So you can see that once you get compromised, there's an entire multi-level of attack, right? So let's take a closer look at some of these barriers. First of all, you got to get in. So some of the physical barriers we have, our skin, for instance, and our mucous membranes. So those are designed to be barriers to intrusion. So for instance, your skin has lots of keratin, which is a tough type of a protein. And basically it creates a tough exterior, a tough hide. And this basically, the outer portions of our skin will naturally slough off. So that's exfoliation. And as they slough off, they'll take all these little bacteria away from the body. And so that's a physical barrier. Mucous membranes is another physical barrier. Ciliated cells tend to basically secrete mucus. Which sticks to pathogens. And then they tend to sweep away that mucus with the pathogens in it. Like, for instance, into your cough zone, if it's a respiratory situation where you can cough it out. Then your chemical barrier is typically when you're secreting chemicals that are antibacterials. For instance, secretions of your sebaceous glands will weaken bacteria on the skin. Sebaceous glands basically create oils and antibiotics, kind of antibiotic molecules. Your sweat, your saliva, your tears all have a lysozyme. These basically will attack bacterial proteins. And thereby chop it up. So basically you're kind of attacking them. So this is your first line of defense, right? So your physical and your chemical barriers. When you take a look at your chemical barriers, the acid also that you're producing from your stomach, inside your stomach, will be a huge antibacterial environment, right? So this inhibits bacteria or just kills it. Also normal flora, that is to say, microbes in our mouth and our intestines. These also prevent potential pathogens from taking up residence. There's basically just no room for somebody new. This is basically, they're outcompeted. By your natural flora. So your own probiotics will actually outcompete the bad guys, and they won't be able to get a foothold. So for instance, this is one of the reasons why we worry about the chronic use of antibiotics, right? So basically you take a lot of antibiotics. Typically what's going to happen is it's going to kill off your natural flora, leading you to more susceptibility to infection, because you're not going to have that competitive environment in your natural flora to beat off this newcomer who's trying to make you sick. The second line of defense is your inflammatory response. So this is your secondary line. And so ultimately what's going to happen in your inflammatory response is you start to deploy your phagocytes. For instance, you start to go after your neutrophils, your macrophages. And so those guys are activated, and they start to go and they engulf all these pathogens and swallow them up and gobble them up. Typically speaking, you have 4 different signs of inflammation: redness, heat, swelling, and pain. Those are the 4. And so typically speaking, regulating and governing some of this are different types of chemical messengers, one of which is called histamine. Several of you may have actually been taking antihistamines as an anti-allergy medication, but generally speaking, histamine is released by damaged tissue, and the cells in damaged tissue, mast cells, for instance. And so what happens is they secrete the histamine, and the histamine will then cause dilation of the capillaries so that they can basically get backup help. So they can basically, it facilitates the transport of more cells, white blood cells in this case, to the site of infection or the site of damage, so they can basically fix things. And this is kind of what it looks like. So here, first of all, you can see you've got an image, a damaged cell. So this damaged cell is basically you're gonna have this puncture. So you can see you got a puncture right here in the blood vessel, you got a puncture right here in the bottom layers of the skin. And here you can see the bacteria coming all the way in. They're starting to invade now. So what's going to happen is these mast cells are going to start releasing histamine, and that histamine is going to go and it's going to start to communicate and dilate that blood cell. It's going to communicate to some of these other phagocytic cells to basically come in and help. The next piece is the macrophages that are already there on patrol, basically will start to attack the pathogens. So here's your first line of attack. So it's going to start attacking those pathogens, and start creating the inflammatory response. And then finally, you're going to do blood clotting. So you're going to plug up that hole so that you don't lose any more blood, and that's going to prevent blood loss. So that's kind of your basic pathway to injury. So the signs of inflammation, so for instance, redness is typically due to the excess blood flow that causes that skin to redden. But because your blood is hot, it also becomes warm. That's the reason why there's local heat. Typically speaking, this is an inhibitory thing because it inhibits the growth of pathogens. So basically it makes them a little more uncomfortable. And then generally speaking, this will increase your blood flow and your white blood cells to that area. And so once you get that increase in blood flow coming to the area, then that'll create swelling. And of course, you have pain receptors in that same area. That's where you feel pain. So with increased permeability of your capillaries, basically this allows those proteins to escape into the tissues, and then your blood clot basically will stop the bleeding. And the excess fluid in the area, because you spilled fluid everywhere, is what causes the swelling. And then generally speaking, the swelling creates pressure on the nerve, and that causes pain. So pain is basically the pressure of the swelling on the nerve. So if you want the pain to go away, put an ice pack on it, and the pain should go away, because you're going to relieve the pressure, reduce the swelling, relieve the pressure, and that'll relieve the pain. So when you take a look at the inflammatory response, the white blood cells we talked about, macrophages, for instance, move out of the bloodstream and into the surrounding tissue. Now, some of the first responders that we have here are neutrophils. These guys are first. So they're phagocytic. They'll go after debris, dead cells, and bacteria. And generally speaking, these will do a good job of containing. So they contain the spread. of the pathogens, localizing it and keeping it small. However, they die off a lot. And they become, in numbers, they become yellow and white bodies that are dead. And this is basically what pus is. So remember, a neutrophil basically kills like a bee. That basically means it kills once and dies. And that's the reason why they form pus, because if they're actively killing on something, then they're just going to be bodies laying all over the place, and that's going to build up at some point at the site of infection. That's going to create pus. So generally speaking, when the neutrophils have a bigger job than they can handle, what they do is they start to secrete their cytokines. These are those chemical messengers that essentially is calling for backup. And as a result, the monocytes, which are in the bloodstream, will move into the tissues. And when they do, they turn into macrophages, which are basically powerful phagocytes. They can basically destroy a lot of different things. And a macrophage is important because a macrophage kills like a wasp. Which basically means it kills many times. Before dying. Just like a wasp can sting you many times before it dies. So some of the protective proteins, so those are some of the cells, the inflammatory response piece. So there's obviously a lot more to that story, but that's where we're going to leave it for now. So some of the protective proteins, complement, for instance. So basically what complement is essentially plasma proteins. These are proteins in your plasma, and typically we kind of designate them with the letter C for complement. And so these guys are going to be involved in a couple of things. First of all, they're involved in the amplification of your inflammation response. So they are on duty and they're active during the inflammation response. Some of them will actually attach to your mast cells that will then trigger the histamine release. So an antihistamine can either work at the level of the histamine or at the level of the complement to prevent its release in the first place. Others will call phagocytes, like macrophages, to the scene, basically getting more people there. And then some will actually form what's called a MAC, a membrane attack complex, which essentially pokes holes in the membrane of the bacteria and bursts it. So once you poke a hole in the bacteria, it just bursts it. This is what it looks like. So here's your mast cell. So you can see your complement proteins will basically insert themselves into the membrane. And as they insert themselves into the membrane, they kind of form these like little open holes. And then this basically allows all the liquid and the fluid of the cell to just essentially leak out. And then it just bursts these cells. So either you can allow the cell to leak out, or in this case, what they're doing is they're loading water fluids, they're pumping fluids into the cell, blowing it up like a water balloon. And then you can see it just burst from the inside out. And this is basically a dead cell. That kind of looks like a barfing cell now when I draw that face on there, but that's basically what happens. So you essentially destroy this by literally poking holes in it and allowing it to burst. Another type of protective protein, which we saw on the previous slides, was called interferons. So interferons are basically chemicals that are produced by cells, and these are typically antiviral in nature. So they basically act as a warning to other cells. So for instance, a virally infected cell will produce an interferon. And this is as a warning to other cells. Stay away from me. I'm infected. So this interferon will bind to receptors on a non-infected cell. So here's a receptor in a non-infected cell. The interferon will come in and bind to that. And then this will basically produce molecules that will inhibit viral replication. So the virus can't grow. So it's like almost like the interferon for the cell acts like, so let me just put negative virus in here. So that's just for that negative effect on the virus. So it's kind of like almost like a vaccination, right? So the interferon sort of vaccinates the normal cell to basically create this antiviral, so it doesn't get attacked by the virus. And so that's kind of what vaccinations do. Now let's take a look at, that was the innate immunity. Now let's take a look at the adaptive immunity. So this basically is another half or another side of your immunity. When the non-specific have failed to prevent the infection, or they're overcome, they're just basically, there's too many, then your adaptive will kick in. And your adaptive is a little bit different because there's a mechanism to remember the way it killed this particular bacteria. And some of these adaptive systems will also be anti-cancer. So this is how you actually screen out cancer cells in your body and eliminate them. Typically speaking, adaptive defenses require a source of recognition, an antigen. This is basically a recognition particle that allows the system to identify the pathogen. So an antigen basically is a recognized particle that's usually a foreign particle, like, for instance, pieces of bacteria, viruses, molds, parasitic worms, things like that. They can all be antigenic. That is to say, they can all display some sort of structure that can be recognized by the immune system to identify it as an abnormal presence. And so typically speaking, since it's based on normality and abnormality, it's not based on good or bad, then we can also use this to screen for instance, abnormal plasma membrane proteins produced by cancer cells. So the way you can screen for proteins by looking for abnormal proteins, basically, that are acting like antigens. So the immune system, the important thing is only its recognition basis is only self versus non-self. It's not good or bad. So you either belong in the body or you don't. And if you don't, you're going to get attacked by the immune system. And if you do, you're going to get a pass. So in the adaptive system, we have two major types of cells, lymphocytes, right? So we have lymphocytes, one called the B cells, which we mentioned earlier. The other ones are the T cells, which we also mentioned earlier. The B cells are the ones that are produced in the bone marrow and mature in the bone marrow. The T cells are the ones that are made in the bone marrow, but then they mature in the thymus. So generally speaking, each of these two groups of cells, the B cells and the T cells, have specific antigen receptors on them to recognize different types of either plasma proteins that bind to particular antigens, or also to abnormal proteins from like bacteria. And so generally speaking, each lymphocyte only has one type of receptor. And so the receptor and the antigen fit together like a lock and key. Like, for instance, if you have a particular receptor for an antigen like this, the antigen on the other thing will actually lock into. So if this is my bacteria, will actually lock into that receptor and it'll recognize it like a lock and a key. Generally speaking, we have two major pathways that are driven by these two populations of cells. So we have what's called cell-mediated, and we have what's called antibody-mediated or humoral. So in the cell-mediated, this is driven by our T cells, which basically kill things on a cell-to-cell basis. It's like cell-to-cell combat. It's like old school warfare, where one warrior had to look another warrior in the face and basically drive the sword into their body. That's kind of what the T cells are doing in cell-mediated. It's cell-to-cell combat. They're identifying the cells, and they're destroying or killing the bad cells. And the antibody-mediated, this is also the humoral. This is basically where the B cells will make antibodies, and the antibodies will actually come in and bind to the pathogen. So this is kind of like long distance attack. The B cells don't actually do any of the attacking. It's almost kind of like artillery fire, right? So the B cells will launch the artillery shells, the antibodies, which will then locate the enemy and then basically blow them up. So it's more like long range warfare. That's what the B cell lineage is like. And so those are your two main types of adaptive. And so this is kind of like where you see these two main types. So first of all, you see your cell-mediated immunity, which essentially has targets the antigens on the surface of cells. So for instance, you start off with what's called an antigen presenting cell. That's basically what APC means. And so generally this can be a macrophage. So macrophages who've been in the trenches gobbling up these bacteria will devour them, chop them all up. They'll take the bacterial proteins as an antigen and display it on their surface so that cell-mediated immunity can see that, screen it. And then once they recognize that antigen, then basically what's going to happen is they're going to start developing T cells to be able to work. And so the two different T cells they make are, first of all, what's called cytotoxic T cells. These guys are the ones that will go after the bad cells and kill them. And they have like regulatory cells called helper T cells. And so these will kind of help coordinate the response, these regulatory T cells. From there, your cytotoxic T cells will go and attack the bad cells, and they'll cause those infected cells to basically blow up. So apoptosis is essentially cell death. It's cell suicide. So these cytotoxic T cells will go and kill or cause to be killed these bad cells or these infected cells. On the other side of things, in adaptive immunity, you have the antibody-mediated immunity. This basically will target usually free antigens. So it'll locate an antigen by antibody because it'll see that because you're circulating through your system. And then once it sees that antigen, then it's going to produce what's called two different types of cells. One of those cells is what's called a plasma cell. This is the one that actually makes antibodies. And these antibodies will basically go and they'll bind to all these different antigens and essentially gobble them up and destroy them. Now, both of these guys will produce what's called memory cells, T and B cells. So these basically remember the profile of the pathogen. So for instance, when this pathogen comes your way again, you already have a memory of it. You already have a profile of it. And so all you can do is just basically cut straight to the chase. You don't have to do any development or screening or identification. You already know who it is, and you can go right after it, and you can basically kill it off. Okay. So, whoops. I think I kind of messed this up. There we go. I think that's it. So those are how the two work. Your B cell receptors typically are the receptors on your B cell lymphocytes. And these guys will be responsible for binding to the antigens. And typically only have one type of B cell receptor. And so this then will produce copies of itself, and then it'll make clones of itself. So you basically, once you figured out the answer, you basically make multiple copies of yourself so you can upregulate the response. So here's kind of what it looks like. So here's your B cell. You have multiple different types of B cells with different types of receptors on it for different types of antigens. They don't all work. So there's kind of like this developmental screening component, right? So this activation piece. So essentially what's happening is you have all these B cell lymphocytes, these receptors that are screening for your correct fit. Once you get the correct fit, it activates that B cell to start going through clonal expansion. You're going to be making lots and lots more B cells. Some of these will become memory cells, and others will become plasma cells, which will then release tons of antibody into the system to go and attack the pathogen. That's what the antibodies are doing here. And this will basically ultimately destroy itself once you're done with the infection, and then you'll lose your plasma cells, but you'll retain these for future infection. This is the reason why on initial infection, it takes a while for us to basically get over it, because it takes a while for the B cells to sort through all of these different receptor options to find the right fit. But then once we get the second infection, we have immunity to it because our immune system already knows what to do with it. So it just cuts straight to the chase. Generally speaking, with your B cells, these guys are going to be making your antibodies. So that's your antibody driven immunity. They're produced and finished and matured in the bone marrow. They will typically recognize only one antigen at a time. And once they do, they'll expand so that you can mount a comprehensive attack. And then they'll release antibodies from their plasma cells that will then attack the pathogen throughout your body. So during clonal expansion, which I think we see up here, the B cells will be releasing these cytokines, or these chemical messengers. And so these are communicated back and forth between the helper T cell, or the helper T cell is this regulatory cell. And it's going to be communicating with the B cells as it's going through its components. So the regulatory T cell is not just regulatory for the T cell lineage, like the cytotoxic T cell, but it's also for the B cell lineage as well. And so ultimately, this is what regulates the expansion and activation of the B cells. And then they basically expand into plasma cells and memory cells. And this basically, the memory cells, gives us our database so that we can maintain long-term immunity. The answer, so to speak, to the problem that is essentially this particular infection. So after the infection, basically what happens is the plasma cells are no longer needed, and so they go through apoptosis and they're destroyed, but we retain those memory cells. Also, the antibodies are also retained in the bloodstream. So this is part of our plasma now. So it basically stays in our bloodstream, and we retain that throughout the rest of life, perhaps, or for a long period of time until we lose immunity. Generally speaking, when you take a look at the structure of an antibody, it has a couple of major parts. First of all, it has what's called heavy and light chain, long and short chains. These are broken up into a couple of different pieces, one of which was called constant regions, so regions of no change. And then, of course, they have areas of these constant regions that are called variable regions. So the V is the variable region. So these variable regions are essentially what allows you to be able to select through and get a correct match to the antigen. So these are basically like all constantly mixing up. In order to find the right match to the antigen. So it's kind of like a random number generator. You don't have the solution to the problem just yet. So what you do is you just basically create all these random variations, and eventually you're going to get a shape here in this variable area that's going to fit that antigen. It's going to bind to that antigen. That's typically on the end of what we refer to as the light chain. So you have the light chain forming one port of the variable, and you have the heavy chain, which is this guy right here. So this guy's going to be the heavy chain, which also forms the other half of the variable. And then the dark blue piece is the constant region. This is the part that doesn't change. So the light blue areas are the regions that are constantly changing, so that you can constantly sort of mix and match, like a random number generator trying to guess a password. And so you just keep on trying, trial and error. And then eventually you're going to get the right one, and you're going to get the right one. And that's going to be the antibody then that gets selected. And then from there it gets activated and then get amplified during expansion. So generally speaking, antibodies can have different structures. So they can be a single Y shape. These are called monomers, or they may have paired, which is dimers. Like for instance, they could have the single Y shape, or they can be the dimer. So this is the dimer. Or they can be pentameters, right? So clusters of Y shapes like this. So they can have different types of shapes to them. But generally speaking, is one of the things that they do is they basically will neutralize the pathogen. So they'll essentially coat viruses, for instance, and toxins that'll just completely take them out of commission, and then they'll be removed. They can also clump stuff up. They can create what's called an immune complex, which will essentially attract white blood cells to the location of the attack, further calling for backup. So when you take a look at antibodies, not all antibodies do the same thing. There's different classes of antibodies. For instance, there's IgG, IgMs, IgA, IgD, and IgE. IgG is typically the major one in the blood. This is the one that we typically think of. And so this is going against pathogens and toxins. So this is typically the one that we normally think of. This is also the one that crosses the placenta to the fetus and the mother and confers temporary immunity for the fetus itself. Then you have IgM, which are pentameters. These are typically the first antibodies to produce that are produced by the newborn. And so they'll oftentimes be the first ones to appear in your blood after an infection kind of begins. And it's also the first to disappear after the infection is over. This is also the one that was responsible for activating that complement system. If you recall from earlier, this is the one that pokes holes in the pathogens. So that's all basically activities of the IgM group. The IgA are monomers or dimers, depending. But basically, these guys are going to be found in body secretions like saliva, tears, mucus, things like that. And so these guys will bind up pathogens so that they can't get into the bloodstream. IgD is typically receptors that are specific to immature B cells before they actually have a chance to sort of grow up and sort of mature into full-fledged B cells. And IgE typically is what you see during like a parasitic worm infection. So if you have parasitic worm infection, these will be increased in number. Also, if you have severe allergic responses, you'll see an increase in IgEs as well. So that basic list is also given to you in the form of a table. For those of you who prefer tables, this is there for your purposes as well. So we have different antibodies that we'll kind of get there in terms of how they're produced. So generally speaking, if we produce antibodies, we can produce what's called either monoclonal or polyclonal. So in a monoclonal antibody production, essentially, these are antibodies that are derived from the same B cell. So there's basically only one source. So that's monoclonal. And so we can make those in the lab, and oftentimes we'll use those to sort of generate antibodies, and we can vaccinate people with antibodies themselves. We also have polyclonal, where you have multiple sources, not just that one B cell source. So this is basically what it looks like when you have monoclonal antibody production. So generally speaking, we're going to go, first of all, from our plasma cells. So we screen for a plasma cell making antibodies that are useful. And then what we do is we basically make use of a myeloma cell, a cancerous myeloma cell, which basically divides at a high rate. And then what we do is we fuse the plasma membrane to the myeloma cell to create what's called the hybridoma. It's a hybrid cell. So it's basically a plasma cell that divides rapidly and produces at a high output. So what happens now is you have a hybridoma that's going to be putting out your antibody of choice, and it's going to be making tons and tons and tons of antibody, and then you can harvest those antibodies. That's basically how we make monoclonal antibodies. Now let's take a look at T cells and cell-mediated immunity. So T cells, you have a T cell receptor, which typically is a unique receptor on the surface of each T cell, where B cells have unique receptors. T cells, however, can't recognize an antigen on their own. So B cells can recognize on their own. However, T cells need the help of an antigen presenting complex. So these basically are the ones that assist T cells in recognition. So if you just have a pathogenic protein floating around on a T cell, they won't be able to recognize it. They have to have it in context with that APC, or that antigen presenting cell. So generally speaking, this APC will phagocytize the pathogen, like for instance, a macrophage. Then they'll travel to the site of the T cells in the lymph node or in the spleen, and then they will display that bacterial pathogenic protein to the T cell itself. Generally speaking, the ingested pathogen parts are broken up by the lysosome, and a piece of that is displayed on the major groove of what's called the major histocompatibility complex. This basically is the self-tag of your cells. So all of your cells have a self-tag. So all of my cells have a tag on it, that the MHC, that basically says, "Hi, I'm Mark's cell." And so this self-tag basically signals the T cell that the APC is normal. But the abnormal protein is abnormal. So this is the reason why T cells need help, because they can't just recognize an abnormal protein. They have to have it displayed in conjunction with this self-tag so that the cell can scan both of it. It can see the normal piece in the MHC1 complex, and then it can see the abnormal piece in the pathogenic protein. And from there, it's able to check down and it's able to basically do its job. Generally speaking, of your self-tags, you have two different types of self-tags: MHC1, class 1, and class 2. And the class 2 proteins, which are typically referred to as HLAs, or human leukocyte antigens, these are basically your self-tags. These are the ones that are in all body cells. And you have three basic groups. You have HLA-A, B, and then of course you have DR, with different types of protein variations on their surface. And what happens is we have, each of us, a unique combination or a profile on our surface of our cells. Our HLA profile of A, B, and DR is unique to us. So that kind of tells me that that's me. So really what we have is this HLA, almost like a barcode, a profile of proteins that's unique to me. And so my cells, by that, say I'm Mark. And so if I have Bob's liver, then his HLA, A, B, and DR profiles will be different. It'll be like a different barcode. And then my immune system sees that and goes on the attack. Identical twins, because these are all genetically derived, are genetically the same. So their HLAs are the same. So for instance, if I was a twin, I could give my kidney to my twin without fear of rejection. So how do T cells then clonally expand once they are presented with an abnormal protein by their APC? So ultimately, you make many copies after your macrophage presents this antigen to the T cell. And generally speaking, it will have a very specific T cell receptor. That's what TCR stands for. And it'll bind to this particular antigen. That process, the binding of that, will then activate the T cell. And that will then cause it to undergo clonal expansion. So it's kind of like a switch. That turns on expansion. So recognition is like a switch that turns on expansion. In some cases, some T cells will become cytotoxic T cells and will go do cell-to-cell combat. In others, they will become helper T cells, in which case they will regulate responses. So it's kind of how it works for clonal expansion. So here you can see you have your different T cell receptors that have been developed in the thymus. And then you're going to have your macrophage. So this is going to be your antigen presenting cell. And it's going to present with your MHC1, that abnormal protein, with your normal MHC1. That basically will select out, almost kind of like you're using a magnet to pick out a needle in a haystack. You're using this as a magnet to select the correct type of T cell receptor. Once that happens, then that turns on clonal expansion, and you start making lots and lots and lots of this T cell. In this case, happens to be a cytotoxic T cell, which will then go after the bad guys. And then once they're done, then they go through apoptosis. They essentially self-destruct, but you retain a memory T cell so that when this happens again, you can respond much quicker. So the cytotoxic T cells, how do they kill? Right? So what is the nature of how they do hand-to-hand combat? Let's take a look at this a little more closely. Inside the cytotoxic T cell are storage vacuoles, and these guys will create a component that they'll have in them enzymes called granzymes, and then another type of protein called perforins. And we'll take a look at the structure of what these guys do here in just a second. So ultimately, when a cytotoxic T cell, like for instance, in a virally infected cell or a tumor cell, both of these will have abnormal structures on the surface. What they do is when they bind to this, that releases these perforins, which punches holes in the plasma membrane, similar to complement. We saw this pattern before, right? Complement poked holes in the pathogens. In this case, cytotoxic T cells with perforins will punch holes in the plasma membrane of the infected cell. They will also deliver these granzymes into the pore itself. And this will basically cause that cell to undergo apoptosis, which is basically cell destruction, cell suicide. And so this way, this cytotoxic T cell can go from cell to cell, determine if you're normal or abnormal, and then basically cause you to destruct. So here's what it looks like. So here's a virally infected cell or an abnormal cell. Typically speaking, what happens is on your MHC, this is your MHC right here, that purple part. So on your MHC complex, what you do is you basically display proteins, both normal proteins and abnormal proteins. So whatever you happen to have going on, you always present a sample of what you have inside of you. In this case, this protein is abnormal. And so when the cytotoxic T cell comes in and sees you, then what happens is it basically kind of locks on to you, and it starts to create these perforins. So here's the perforins it's releasing. And basically, as it releases those perforins, it's going to basically sort of insert into the membrane, creating this open channel, this open pore, very similar to the way complement insert in the pathogen. And then once you have this opening here, then you start to turn in the enzyme, those granzymes, which will then cause you to go through apoptosis, which is cell death. Okay, and your cell disintegrates at that point. And so that's basically how your cytotoxic T cell goes. So here's a great scanning electron micrograph. So you can see your cytotoxic T cell, TC right here, attacking what is some sort of a target cell up here. And so basically it's attacking this virally infected cell, and then that cell is going to basically self-destruct here in a little bit. Okay, so that's cytotoxic T cells. What about helper T cells? So helper T cells, basically what these guys do is they'll secrete cytokines, right? These are those chemical messengers. And so what they'll do is they'll basically enhance the response time and regulate the response of all the immune cells. For instance, they will help to not just regulate B cells, but also T cells. And so as a matter of fact, B cells can't be activated without your T cell help. So they need the helper Ts or TH. Now, the reason why AIDS, or HIV rather, is so deadly is because this virus actually infects the helper T cells. So you're basically taking out the regulator that pulls all these responses together. So if you take a look at helper T cells basically regulate the B cell lineage, but they also regulate the cytotoxic T cell lineage. If you take out that helper T cell, then you're also taking out your adaptive immunity. By basically destroying helper Ts, destroys your adaptive immunity. And basically, if you have complete immune system collapse, that's basically full-blown AIDS. So some of the characteristics of our T cells, for instance, typically cell-mediated, will give us immunity against virally infected cells and cancer cells by screening their abnormal proteins on their surface. These guys are going to be produced in the bone marrow, but they mature in the thymus, unlike B cells, which mature in the bone marrow. Cytotoxic Ts will basically go after non-self antigen bearing cells, including cancerous and viral cells. And helper T cells will regulate the response by secreting cytokines and controlling how each of these are regulated. Okay, so now let's go ahead and take a look at sort of some vaccination issues. And so basically how we get immunity. So we have two different types of immunity. So we have passive immunity and we have acquired immunity, or excuse me, active immunity rather. And so these are acquired immunity. So this is what we build up over time. So immunity can be broken into two different types. You have active and you have passive. So let's take a look at active immunity first. Active immunity is basically when your body actually makes antibodies against a particular antigen. So it has to work for it, has to fight an infection of sorts in order to generate its antibodies. So this can be through natural infection or it can be through vaccinations. So we can basically give your immune system a little bit of an antigen to fight against. And so it's kind of war games. It'll fight against these. The vaccination will contain like a dead virus or something like that, or a pathogen that's dead, and then allows your immune system to fight against it, and then it'll develop immunity to it. Typically speaking, when you have first exposure to a new antigen, it's what's called primary response. It takes a little bit longer to get over it at that point. And then the second exposure is called secondary response. Usually that one moves a little quicker. And this depends on the nature of the memory of the lymphocytes, right? So if the memory cells or the memory lymphocytes don't last long enough, then you need to get a kind of a memory jogger. You have to remind your immune system of its immunity. And so that's what a booster is. So that's kind of what it looks like. So basically your plasma antibody concentration on first exposure, primary exposure, you'll basically have very low amounts of antibody. You can see you peak after about 20 days or so. And then after about 30 days, you can see your primary response starts to crater. And so after about 2 months, it's largely back down to zero. Then if you do a second exposure in a couple of months, then you get a much more aggressive response. So what you're doing here is you're burning that memory into your immune system by doing the secondary response. It's kind of like when you study to saturation and then you just kind of put everything away, you go walk away, go for a walk, clear your mind, you come back and you hit it again. You retain more information. So that's like secondary response. And this is basically happening around 90 days. And then notice it basically doesn't go down to zero. It basically retains some sense of memory and some sense of immunity in terms of the antibodies that are retained in your plasma. So that basically is active immunity. Passive immunity is essentially when you are given antibodies to combat a disease. Your body's not producing it. Typically you're given it, it's given to you. So unfortunately, passive immunity is only temporary. So you're not going to hang on to this one for a long time. For instance, a good example of passive immunity is when a newborn infant is getting antibodies from that cross the placenta from the mother's blood. Also breastfeeding, right? So breastfeeding has a lot of antibodies in the breast milk. And these antibodies will go into the baby and basically provide some low-level coverage for the baby while the baby's own immune system is trying to get itself ramped up. You can also receive antibodies by injection, by vaccination. And so this is kind of what it looks like. So here's your placental crossing of the mother's antibodies from her bloodstream going through the placenta and then basically covering the baby to make sure that the baby is safe. And then in breast milk, the breast milk part of it will be harboring the antibodies, which will then go into the baby and give the baby some coverage short term while it's still feeding and its own immune system is still getting up and regulated. And then we can also vaccinate with antibodies as well for those of us who are already adults. As a matter of fact, that was one of the strategies that we used in COVID was antibody vaccinations. This is what's happening if you're getting a plasma injection. So typically with plasma, that's where your antibodies are. If you just basically harvest plasma and inject somebody with plasma, you're going to be getting their antibodies. And that'll give you short-term coverage. But if your immune system didn't make those antibodies in the first place, it's only going to be temporary. At some point, your immune system has to be able to make its own. Now let's take a look at cytokines and immunity, right? So cytokines are important because this is the communication system. So basically, cytokines are signaling molecules that are essentially produced by the T cells and macrophages and other cells. And this is basically how they regulate their functions. So they regulate their formation and their function. So they're basically constantly talking back and forth to each other. For instance, interferons and interleukins are types of cytokines that are used in different types of medical treatments. For instance, interferons we already met, and interleukins are a different type of white blood cell communication, as communication between white blood cells are talking back and forth to each other. Now let's take a look at a couple of disorders, right? So first of all, HIV/AIDS. So HIV or acquired immune deficiency syndrome, which is AIDS, there's basically two types of HIV. There's HIV-1, which is the most common, and there's HIV-2. HIV-2 is typically found in the green monkey in Western Africa, and HIV-1 is basically a replica that's found in chimpanzees, and they're common in West Central Africa. So what this suggests is that the HIV virus originated in non-human primates and then crossed over, mutated, and then was able to move into humans. Oftentimes, probably as humans ate non-human primate meat, that's the most likely way that it actually crossed over into humans. And then from there, it's able to spread through multiple mechanisms, including sexual contact. So HIV, remember, is a cell with a particular cell surface receptor on it. And so what it goes after is the helper T cells. And so it destroys those, and it also destroys macrophages, both very critically important cells, right? Because helper T cells are at the center of your adaptive immune system, and macrophages are at the center of your innate. So both sides of your immune system are being attacked by HIV. And so ultimately, the ability to fight this infection is declined. Why? Because you're going after the very cell that you need in order to fight viral infection. And so typically speaking, once the virus spreads and it starts to collapse, then what you get is AIDS, acquired immune deficiency syndrome. So this is typically associated with the collapse of the immune system due to helper T destruction. And so generally speaking, oftentimes what an individual with AIDS dies of is usually some sort of an opportunistic infection, something that's rare, right? That wouldn't happen in a healthy individual. Like Kaposi sarcoma, for instance, is the famous one from Philadelphia, the movie Philadelphia, right? That's something that just doesn't happen in somebody with a functional immune system. We have AIDS patients that are dying of things like the common cold or just like a mild flu, because they have no immune system to fight against it anymore. And then basically they end up dying of these opportunistic types of infections. And so this is kind of the stages of infection. So basically, here's your helper T cell count, which is in blue, and the HIV viral load is in red. So in the first couple of months, the load increases dramatically. And so ultimately, the HIV count goes way up. And then as soon as it goes up, you notice the T cells go down. And so at some point, the load will go back down. So this is usually because of immune system activity. But then as the T cell slowly crashes over time, the viral load will slowly increase over time, and it overtakes the T cell population. And then eventually what happens is once it crashes, then your viral load goes up, and then that basically just absolutely just craters um, your immune system. And so oftentimes a lot of folks are in this sort of chronic phase where they're taking any number of sorts of medications to combat, to keep the HIV load from overtaking the helper T load, and doing everything they can to help their helper T cell population to stay robust so that they can keep their immune system. So ultimately, then we also have, in addition to this, so that was the HIV story. So another thing that we have is hypersensitivity reactions. So this is a situation when your immune system overreacts to a particular cause, like allergies, for instance. Like pollen is not dangerous, right? But that's going to trigger a response in your immune system because it looks foreign, it looks dangerous. So it's going to go on the attack. And so we have allergies, generally speaking, different types of allergens like pollen, food, or whatever. And so this immediate response is typically an inflammation response. Usually what will happen is you'll see the IgE antibodies, which will basically attach to different receptors on mast cells and basophil cells, which will then increase histamine, which will then dilate those blood cells. Ultimately, the histamine will stimulate mucous membranes of the nose and the eyes. So this creates fluid and runny nose and hay fever. So that's basically where this comes from, because you're trying to get the pollen out. So that's what's happening. However, this dilation is also a dilator of airways. So you can actually constrict your airways with histamine. And that'll cause wheezing in severe cases. Food allergies will do a similar sort of thing, only your stomach will respond by nausea and vomiting and diarrhea. So they tend to be more digestion in nature. And so generally speaking, when you have a very severe allergic reaction, that's anaphylactic shock. So that's a situation where you have a really, really rapid, quick response. This is basically when the antigen enters the bloodstream. It's a very rapid and severe response. Not just sneezy, it's very, very sudden, typically life-threatening. It can start causing all sorts of negative parameters, like, for instance, dropping blood pressure due to the dilation of your blood vessels through histamine. Generally speaking, taking epinephrine can counteract this reaction. So those people who have anaphylactic shock risk will carry an EpiPen with them in case they run afoul of this. A good example of this are some of the more severe food allergies, like, for instance, peanut allergies. Nut allergies tend to be very aggressive and can be anaphylactic at times. And so ultimately what happens is you have kind of also a delayed version of this, not just anaphylactic, but kind of a delayed version where you have an irritation, like poison ivy or poison oak is a good example, where you have kind of a delayed response where it doesn't like get you right away. It kind of builds up and then it kind of gets you a little bit kind of on the back end, and it kind of irritates your skin or whatever. So there are other types of immune problems we have, like, for instance, rejection of tissue. That's because you have a violation of self versus not self. So I've got Bob's HLA profile on his liver, but it's not mine. So my immune system attacks it. And so the cytotoxic T cells will see that as foreign, and so they'll go on the attack. In order to keep this, we have to typically take immunosuppressive drugs in order to keep our immune system from attacking our liver transplant, so that we can make sure we have this liver in place for us to be able to survive, and so forth. And so there's a lot of different types of immunosuppressive drugs. A donor or a heart transplant patient or any kind of an organ transplant patient typically has a lifelong trajectory ahead of them of these immunosuppressive drugs to keep them from rejection. So xenotransplantation basically is transplanting from an animal. And so even some organs can be grown in labs. So we're doing more and more sophisticated versions of organ transplant that's a little bit better. So SCIDS, which is severe combined immunodeficiency, is basically where your antibody and cell mediated are not good enough. So they're weak. So it's a weak attack. Typically, it can be either minor or it can be fatal. It depends on the individual. Usually, we'll treat this with bone marrow transplants and gene therapy, which I hear bone marrow transplants can be quite painful. But if you have it, then it's definitely something you want to do. We already talked a little bit about AIDS, right? So this is basically the full-blown portion, the complete immune collapse that's associated with HIV. And so you have a higher susceptibility to risks and autoimmune diseases. These are important ones. A lot of people have questions about autoimmune diseases. Autoimmune diseases are essentially when both your T cells and your antibodies will attack your body's own cells. So it's basically a failure of that self versus non-self recognition. And sometimes it can be environmental, and sometimes it can be genetic. Sometimes it can follow an infection, for instance, rheumatic fever, which is a very, very bad fever to get. Ultimately, this is going to be strep, a type of strep, systemic strep. And then what happens is the strep bacteria, streptococcal bacteria infection, which typically is of the throat, will ultimately actually react with the heart muscle, and it damages the heart muscle and the valves. So basically does lasting damage. It's the reason why it's very dangerous. This is what we think killed Mozart. So those of you who love Mozart, he died young. I think he was like 35, 38, something like that. And he died of cardiac failure. And the reason he died of cardiac failure is because he had many bouts as a kid of rheumatic fever. Some were quite bad, actually. And that probably left lasting damage on his heart muscle and on his valves. And that probably caught up with him in his mid-30s. And then, of course, we had rheumatoid arthritis, which are basically antibodies that are attacking the joints themselves. And so it's kind of what arthritis looks like. So you can see you get disfigurement of the joints. Usually not to be expected until you get older. So usually somebody who's younger and who's healthy isn't going to have rheumatoid arthritis. That's usually something associated with just aging process and the general wear and tear and the breakdown of the body as we get older. SLE, so systemic lupus erythematosus, or just generally lupus. This is essentially when you have facial rash, fever, joint pain, things like that. This is a central nervous system situation. It'll can also attack the heart and the kidneys, and it can be fatal. So this is important because you're attacking some major core systems here that are really important. And so it actually goes after your DNA, which is not a good thing to do. So antibodies, which are actually seeing your DNA as an antigen. And so, except for red blood cells, which don't contain DNA, so this basically will go through a lot of symptoms and attacks DNA wherever it finds it, pretty much in all cells. So all human cells pretty much have DNA. Myasthenia gravis is a situation where your antibodies are going after muscles. So your antibodies will attack the muscles, and it causes severe muscle weakness. Could also create death if it's respiratory failure. This is basically weakness of the diaphragm. And so that could be caused by myasthenia gravis. Multiple sclerosis, which is basically where your T cells will attack the myelin sheath, which is basically in the neurons. So it causes neurons, which then causes misfiring. Of the cells, of the nerve cells, so that you don't have coordinated firing of the neurons anymore because they're unmyelinated. It's kind of like an uninsulated wire, right? Where the electricity is kind of spitting off of this uninsulated wire in kind of random, uncoordinated fashion. The reason why we have insulation on the wire is to give us coordinated flow of that electricity through the wire itself. And so this will cause massive dysfunction. It gets worse and worse, double vision, muscular weakness, muscular dysfunction, or dysregulation. It could be multiple different things. We're still doing a lot of research on multiple sclerosis. Could be autoimmune in nature. We haven't located a specific antigen necessarily, but we do treat it with different types of drugs to kind of slow down the immune response, give it a little bit less aggression. Okay, so that right there is chapter 7. That's lymph and immune. And then next up will be chapter 8.